So good morning, everyone. My name is Shimei Lok. I'm the Provost Chair Professor from the Duke NUS Medical School. So the first speaker for this morning is Li Hanqi. She's a PhD student at the Cambridge Institute of Medical Research. Her research interests include virology and host pathogen interactions. Hanqi?
Morning, everyone. I'm Hanqi, a PhD student in Professor Mike Weeks' lab at Cambridge Institute for Medical Research. And many thanks for having this opportunity to talk about my research on human cytomegalovirus. Thank you. So what is human cytomegalovirus? Well, hear this word, you might think, cyto, something sounds like a cell, and megalo, something sounds like a very big or giant thing. Then you are right. Human cytomegalovirus is a very big and complex double-stranded DNA virus with 170 genes. And of course, from its name, it's from the human cells. In fact, the human cells. So here's a picture showing the cells infected by the HCMV. And we can see that this virus occupies about half of the cells here, and also it can be spread between the cells very quickly. Actually, there are 60% of all of us in this room has been infected by the HCMV. However, it's normally kept silent in our bodies, don't worry about that. However, in the immunocompromised people, it can cause a substantial morbidity and mortality. So it's like a very high infection in the immunocompromised people, such as the people with the AIDS or the organ transplant recipients. Only a few people realize that the HCMV can affect 1 in 100 pregnancies and can cause the hearing loss and intellectual disabilities in newborns. However, there are only 4 anti-CMV drugs available, and all of them have significant toxicities and problems with drug resistance, and also there's presently no vaccine available for HCMV. So how this virus be so successful in defeating our cells? Actually, HCMV is a master regulator of our cell's defensive system. And one particular strategy it uses is to destroy certain cellular proteins. And the proteins are the cellular components responsible for almost every function in the cells. However, there are over 130 host proteins can be degraded or destroyed by the HCMV during the whole course of infection. And this is generally by the interaction of a viral factor shown here and its host target, followed by the recruitment of the waste dumps to remove the damaged part. And inhibiting this kind of interaction using the small molecules may form the basis of the new antiviral therapies. So the question is, how could we prioritize which cellular and viral proteins to study in more detail to give us this kind of antiviral therapies? We have recently established that the host factors of key functional importance can be destroyed during the early HCMV infection. And how could we measure this? The Weeks lab previously developed novel global approaches to identify individual proteins degraded to the fastest and also to the greatest extent during the HCMV infection. And this is all based on a technique called the proteomics, which enables more than half of all cellular proteins from different conditions can be measured in the same experiment. And these conditions normally include adding the waste dump inhibitors to rescue the protein from destruction, or adding the metabolic pause to measure the rate of the protein destruction during HCMV infection. And also, at the same time, the RNA, the transcript of the protein, can be harvested together and to be quantified using the transcriptomics. And here's some data of a very good hit called the HLTF, which our lab has shown it to inhibit the HCMV's replication, so the virus needs to get rid of it. This protein can be rescued in the HCMV infection by the waste dump inhibitor shown here, and also it has a faster rate of destruction during HCMV infection compared to the uninfected cells shown as smoke like here. And comparing the abundance of this protein and its corresponding RNA transcript, we found that the protein was decreased, but the RNA was not. And this indicates that it is the HCMV to destroy this protein instead of regulating its generation. So overall, our lab has shortlisted 35 proteins that destroyed by HCMV during early infection to the fastest and also to the greatest extent. And in addition to HLTF and another factor that our lab recently published, One very interesting hit that has been unexplored so far is DMXL1. So what is DMXL1? Actually, it's a very large protein formed by 3,000 amino acids. And also, it interacts with and stimulates the VTPase, which pumps the protons. So the stimulation of VTPase by the DMXL1 can lead to an acidification of the intracellular membrane bond compartment in the cells, which is called vesicles, in particular, the lysosomes, which will be very important for the functional acid I will talk about later. So now, we want to address how and why does HCMV target DMXL1 for destruction. So first question, which viral factors are involved and which host machinery is required? To understand which viral factors are involved, our collaborator has generated a series of block-deleted virus, each deleted with a block of the viral genome that's non-essential for viral replication. And this clearly identifies that the US29 to 34A block of the viral gene is necessary for the DMXL1 degradation. So when this block of the gene is deleted, the DMXL1 level go back. Then we want to understand which factor in this block is necessary. So we did a single gene block deleted virus screening. So this block, this single gene deleted series of virus encompassing every factor in this block. And this showed that the US33A gene, the virus, the protein of this virus, is responsible for DMXL1 destruction. So when this gene is deleted, the DMXL1 level go back. So what is US33A? Surprisingly, it's a very small protein, only formed by 57 amino acids. And predicted by AlphaFold, we can see that it has a central alpha helix, but otherwise with unstructured region on either side. And its transcript analysis shows that this protein was expressed during the early infection as what we expected. So how this small protein can destroy the 3,000 amino acid DMXL1? We did an interactome analysis. So each of 170 HCMV genes were tagged with a 14 amino acid residue called the V5, and they were expressed in the cells. So the interactors of this gene were trapped with the anti-V5 antibody bound to the base, and then they were analyzed for the interaction. However, this kind of reaction normally gives us like a list of the interactors up to 1,000 proteins, but only minority of them are true. And this was reviewed using based on the computationally machine learning strategy. And conveniently, the two highest confidence interactors of the US33A are RNF123 and UBAC1, which are the two components of the KPC complex. This is a key mediator of the protein destruction in a waste dump called the proteasome. So now we wanted to ask whether the RNF123 and UBAC1, the KPC complex, is necessary for DMXL1 degradation. We used the CRISPR technology to knock out the RNF123 or UBAC1 or both in the cells, and then infected them with the wild type or US33A deleted virus. So let's see a picture of the DMXL1 abundance in the unmodified cells first. So we can see that the wild type HCMV clearly destroys the DMXL1. However, the US33 deleted virus could not. Then, when it happened in the knockout cells, we found that either the RNF123 single knockout or the UBAC1 single knockout or the double knockout, we found the wild type HCMV could not degrade DMXL1, and it behaves similarly with the US33A deleted virus. And this shows that the US33A destroys DMXL1 using the KPC complex. So now we wanted to understand why. Based on its known function, the loss of the DMXL1 might lead to a less acidic lysosome. So we measured the lysosomal pH in the cells, so the control cells, and also the cells expressing the viral gene called the US33A. And we stained their lysosome using the lysotracker to show its lysosome pH. This is exactly what we found. The US33A cells got significantly lower lysotracker staining compared to the control cells. And this suggested that the DMXL1's destruction by the US33A can lead to a less acidic lysosome. So what will happen if the lysosome is no longer acidic? I want to introduce you a process called autophagy. It is a very important recycling mechanism in the cells to remove or to reuse the damaged parts into the new part. So the autophagosomes can engulf the damaged or defunct cellular materials and then fused with an acidic lysosome. This acidic environment allows the proteins to be chopped up into the component amino acid or the membranes into the fatty acids, and then to allow them to be recycled and reused into the new parts. So does DMX1 destruction impact autophagy? We measured this using a marker of the lysosome called the LC32, and this can be destroyed together with the autophagic cargo in this acidic environment, the autolysosome. So when the autophagy is inhibited, the LC32 level will be increased. And this is what we found. The LC32 level in the US33A expressing cells significantly increased compared to the control. And this suggested that the autophagy was inhibited by the DMX1 destruction. We want to look at that in more detail. Which stage of autophagy was inhibited? We measured this using the LC3, which is the autophagism marker, and the marker of the lysosome, which is called the LAMP1. If the autophagic cargo degradation was inhibited, there will be more LC3 co-localized with the LAMP1, so all things are stored here. However, if the autophagism lysosome fusion was inhibited, the LC32 will be separated more with the LAMP1. And here's the result. We found that in the US33 expressing cells, when the DMXL1 was destroyed, the LC32 was largely co-localized with the LAMP1. So here, so the red plus the green bit to become this kind of like orange or yellow color. And this suggested that the DMX1 destruction inhibits the autophagic cargo degradation. So now, finally, we want to ask why virus want to do that. It's the DMX1 destruction, but the virus can benefit the virus itself. So what we do is we infect the cells using the wild-type HCMV and also the US33A deleted HCMV. And then we measured this virus associated with the cells and also the virus released from the cells. And here is what we found. The US33A deletion from the virus significantly reduced the proportion of the cell-associated virus. This suggested that the DMX1 destruction by the HCMV might enhance the viral intracellular survival, and this might enhance the cell-to-cell spread of the virus during the in vivo infection, so when the virus infects our body. So overall, in conclusion, what I found is that the US33A destroys DMXL1 by redirecting the cellular KPC complex. And DMXL1 destruction inhibits lysosomal acidification and the recycling of the autophagic cargo. And DMXL1 destruction enables the increased the cell-associated HCMV and might help its intracellular survival and cell-to-cell spread. And targeting the DMXL1 destruction by US33A is our future direction. So we will use this, and it might be a very good new antiviral therapies. So at the end, I would like to say many thanks to all the Weeks lab, especially my supervisor, Mike Weeks, and our alumnus, Alice Fletcher Etherington. And also I would like to say many thanks to the Rubinstein lab in Cambridge, and which helps us a lot for our autophagy assay, and our virus collaborator in Cardiff University, UK. And thank you very much for listening, and I'm happy to take any questions. Thank you.
Hi, very interesting talk. So I have two questions. The first one, I was just wondering, so how essential is the DMXL1 degradation for the virus infection to work? So do you have any assays or experiments where you can artificially block DMXL1 degradation and still look at what happens to the viral infection?
So you mean they in vivo or something?
No, so you're looking at the degradation of DMXL1,
yeah,
and that's correlated with, say, viral infection. But if, say, in a scenario where the viral infection is still there and DMXL1 is not degraded, does that make a difference? Is there any way to test it?
Well, thank you. That's a very good question. Yeah, when the virus infects the cells, the DMX is still there. But I mean, when we knock out the US33A, the factor that degrades DMXL1, and we found the virus that associated with the cells significantly reduced. So that's but the virus are still there. Yes, you're right. So it's a kind of one factor of that. But yeah, because the virus is very complex stuff, it also can regulate the other factors of the cells. So yeah, you're right. It's not the only single important things in the cells. It's never be. Yeah, but
what if you do it in an overexpression model of DMXL?
Overexpression of DMXL1 to...
Yeah, so would the virus still degrade all of the DMXL1?
Yeah, because it's like, when we infect the cells using the virus, we found that, when the US33 deleted, the DMXL1 level got back to like 34-fold, which is far more than the normal expression level of the DMXL1. So we think the virus is very sufficient to degrade that. But you're correct, yeah. I think later maybe a overexpression of the DMX1 is worth to try. Thank you.
Great, thanks. So the other question, so when you delete the S33A or overexpress it in the virus, it probably has other effects than just degrading DMXL1, right? So for those experiments, you have a control to show that the phenotypes you see are DMXL1 degradation specific and not just a general effect of knocking out S33.
Knocking of the... Oh yeah, so it's like we got another virus also to show this kind of thing. So we're not only using the US33A overexpressing cells for sure, and we're also using the adenovirus, and we saw exactly the same results shown here. And also we infected the cells with the wild type and also US33 deleted virus, and we saw another way around. So it's also can prove this, yeah, true. But here I just only showed the US33 expressing cells.
Right, great. Thank you.
No worries.
Hello, good morning. So first, thank you very much for your very good presentation. So I just want to ask a very simple question. So based from your conclusions, what could be your future directions on your research? Thank you very much.
The future directions of this
Yes.
viruses, so we will firstly prove it. Whether this kind of DMX1 destruction can enhance the cell-to-cell spread of the virus. And we have a virus, actually, we already got that. It doesn't have the US33, and also it's only spread between the cells, so not released from the cells. And we'll do this one first. And also, it's like in the future, if we can collaborate with some very clever or intelligent people doing the kind of drug design, and also doing the kind of AI and computational simulation of the disruption of this kind of protein-protein interaction, so we can futurely try to design kind of
Thank you very much. Good luck and congratulations.
Thank you.
Thank you for the talks. So I have two questions. The first one is that, is DMXL1 expressed in every cells? And the second question is that, is this mechanism preserved in different type of cells, particularly like the immune cell and the brain cells?
Yeah, so the DMX1 is expressed in most of the cells. So like the HeLa cells also is not and the kidney cells and epithelial cells, they're all there, but the expression level was relatively low in all the cells, including the cells we use, the fibroblast. And another thing is sorry, would you like to repeat your second question?
Yeah, is this mechanism preserved and conserved in different type of cells, particularly immune and brain cells?
We can try to figure this out. It's because some of the neuron cells is permissive for the HCMV infection, so we can try that in this kind of cells. But because of the the restricted cell type that HCMV can infect, so we only use the fibroblast. But in the future, that's a kind of direction to look at if it's kind of general thing. Thank you.
Thank you.
I have questions. So a lot of viruses, they use endosomes to enter the cells. And also when they matures in the cell, they use also vesicles that are low in pH.
Yeah.
So CMV doesn't use any of those?
When CMV enters the cells, it doesn't use the endosomes to do that. Sorry.
So it doesn't need that?
Yeah, it can get into the nucleus very quickly, within the 12 hours. It can also spread the kind of very early genes to start its replication. And way to get out, there is some evidence showing that exactly the the autophagism, it can help the cells to egress with it from the cells. There are some literature already shown that. So when the cells getting out from the cell, it takes some bit of the autophagism. So that's why maybe when the DMX1 destroyed, it can reduce the cell release as well. And also the thing is that normally HCMV, when it's getting out, it's using this kind of acidic environment to assemble its thing, but you can see it's like not assembled in one single endosome. When I show the infected cells, they're like the particles is kind of so messy in the cells, it's just distributed.
So in induced autophagy, so does it mean that the cell will die?
No, this autophagy is a kind of like recycling thing. So it can be associated with the cell death before, but actually autophagy is kind of like enhance the cells to live further. So they recycle the damaged part and the old parts to try to promote the cell death instead of like inducing the cell death.
Okay. And one last question. So you use a drug to screen for the host target, right, that is bound by the viral protein.
Yeah.
So did you look at whether the drug actually binds to DMXL1 directly?
So actually, we didn't look at that. Yes, you're right. Yeah, actually, because we use the drug as get into the with the DOMs such as the lysosome and the proteasome, but we didn't look at if it interacts.
So you don't know the mechanism of inhibition by the drug?
Inhibition of
the mechanism, how it does this inhibition.
How?
So the drug inhibits the infection, right?
We didn't have any drug to inhibit.
I thought you used a drug in order to get the mass spec.
Oh yeah, that's why it's like it's not. So the drug was applied before the infection, so it's just to get into the proteasome. So it's MG132 and lupeptin. So it inhibits these kind of like proteasome and lysosome cell environments to allow the proteins not degraded that. So it might not affect the infection stuff, I think.
Oh, okay. All right. I think that's it then. Okay. Thank you, Hanqi.
Thank you.